Okay, let's move on now to DNA fundamentals where we were before. So in DNA, we learned last time that the base is thymine with the adenine AT, and this is G, guanine and cytosine, so GC. So AT make 2 hydrogen bonds, and then GC make 3 hydrogen bonds, and that's how DNA is connected with one another. Another important thing which I missed last time is that DNA, whenever you look at the sequence, it's always mentioned 5 prime and also 3 prime ends. So whenever there is a segment, let's say ATGCC, so always written some at one side 5 prime and the other side 3 prime. For example, if we look at this one, although this is not a great structure, so let me go to the next page. So you see this one is written 3 prime, right? And then on the opposite of that would be 5 prime. So there are two different. Do you know why they are named 5 prime and 3 prime?
The direction they transcribe.
Yeah, normally whenever there's an elongation in a PCR cycle, which we will learn later on what is PCR, but basically when we synthesize the DNA from one single strand to double strand by using a polymer chain reaction, so then it elongates towards the 3 prime, not towards the 5 prime. So 5 prime we cannot elongate it. So but for it normally elongates from 3 prime to onwards. So but why we call it 3 prime? Why not 2 prime? The answer is here actually somewhere.
Does it deal with the structure, like the atom, the specific atom on the ring?
Yeah. So if you remember the pentose sugar we talk about, so this is a one sugar, pentose. So this is a C1, C2, C3, C4, C5. So, and you can look at it that, let's say if you take this example, this phosphate group PO4, at one side it is attached with C5, and the other sugar group it is attached with C3. So basically phosphate group is making a bond between C5 and C3 side, right? So see, it's this naming trend, so we just named it this one as a C3. If we start from here as a C1, C2, C3, C4, C5. So basically it's connecting C3 and C5 and C3. And that's why one end is called, so let's say at this side we have C5, so this is a 5 prime end, and we cannot elongate on this side. So what we normally do is normally elongation happen on this side, which is 3 prime, and in this 3 prime end what happens, a new sugar phosphate group can come here, it can make a bond with OH right here, and then the other, because it is minus 3 I believe, then the second it will also make a bond with another sugar group which is like this towards the C5. So but the last sugar on this side would be the one where 3 dash end would not be bound with the PO4. So basically it's the direction, it's a naming based on where phosphate group is attached. So this would be called 5 prime end, this will be called 3 prime end. And the DNA strands actually runs in parallel in opposite direction. So basically if this is a 5 prime, and if it makes a complementary structure, so then the other 5 prime would be totally opposite to that. Okay? So if 5 prime is here, then this 5 prime will be on this side and 3 prime will be on the top. So they run in anti-parallel, I would say. And what else? And the hydrogen bonding, we did talk about it briefly previously, right? So, but what happens in the hydrogen bonding, whether it's oxygen or, let me turn on the dock camera. So we have different kind of bonds. So one are ionic bonds. So for example, in ionic bonds we have NaCl, right? So let's say we have Na, which is one electron, right, on the outer shell from group 1. So Cl, which is quite a few valence shells, but in the outer shell, how many electrons it has? 5, 7. Okay, so what happens when the sodium and chlorine come close to one another? It makes NaCl, which is salt. Have you ever seen any sodium? How reactive it is? No, we might not have seen it. But I have seen it in some YouTube videos where they were putting pure sodium inside of water, and there was a flame all of a sudden right away. So it was so vigorously reacting with the water and other things. So magnesium, sodium, so they are very super reactive, and that's why normally we don't see them in our environment as in atomic form or as just a sodium. So mostly they have already made some ionic or other bonds with something so to become stable. So but in this case, and the same way chlorine, if it is present in a single molecule, it's a super reactive, and this chlorine is used in a we have discussed it. Do you remember where we have discussed the chlorine is used? In etching, in dry etching, chlorine is used. Okay, chlorine, those gases are used in dry etching process when we want to etch silicon dioxide, glass, or other things. So in this case, so it has one extra electron outside. It needs one electron. So what happens that it can give it actually electron, right? And then it becomes 8. Then its valence should also have 2. So it will be complete. So basically, this will be ionic bond. Ionic bond is the one where the electron is actually exchanged from one atom to other. Now we have another example, which is a covalent bond, right? So covalent is when we share electrons between two atoms. Let's take a simple example of a carbon or silicon. Let's take an example of a carbon, for example. So one carbon atom has actually 4 electrons in the outer shell. The other carbon has also 4 in the outer shell. So what happens that it needs a 4 electron, or it can actually give 4 electrons. It needs 8 electrons in total. So what it does is actually shares 1 electron, come close and make a bond. So basically share 1 electron between the neighboring atom. But how you can share this one with that one? Is it possible? Can it try? Maybe not. I can put another carbon there, right? Like this. So there are actually 4 carbon atoms on the side. One is sharing 1. This is also sharing with this one. There will be another one here. So basically 4 atoms are shared with 4 other atoms. So that's how the carbon is structured. And this is called covalent bonding. So this can also count. This can count that it has a 1, 2, 3, 4, 5, 6, 7, 8 electrons, and this can also count 8 electrons once it has all other 3 ones. But now there are some bonding which is called we call it a hydrogen bonding, right? And that's also very important because in water we have a hydrogen bonding, which is a very common material to see. But in general, in DNA we also have a hydrogen bonding. In DNA. First, in water, what happens that, let's say we have it's H2O, right? So we have oxygen, and then it has 2H. And this is a property when actually hydrogen, when I say hydrogen bonding, it means that when strongly electronegative atom, which can be oxygen, which can be chlorine or nitrogen. So those atoms, when they make a bond with the smaller size atoms like hydrogen, for example, which is very small. So there is an effect. It's called the shifting effect. What happens that because it is sharing 1 electron, so there are actually 2 electrons which are shared. This pair of electrons shared on this side and also shared on this side. Okay, so what happens because of this is a larger atom. So it attracts, this pair is shifted a little bit towards the oxygen. So basically, instead of being here, it shifts a little bit towards this side. So on the oxygen, there is a minus partial charge comes in. So it's not really a full charge, but slight charge. That's why we call it a partial. And then on the hydrogen, partial positive charge means slightly positive. It becomes slightly positive. And that's because of the presence of a very large atom along with the smaller one. And we see that effect. And what happens, let's say now if we have another oxygen atom with the 2 hydrogen, this is H plus, this is minus S, right? This is plus S again. So if all these atoms are present close to one another, so then this plus S will start showing some attraction towards this minus S, right? And it will happen. So it will become oxygen, hydrogen, and oxygen, right? So this is plus S, minus S. So with the other molecules, it start showing some attraction. And that attraction is because of a hydrogen bonding, because it has a partial charge. And now we can think about, if we now look at this structure of a DNA, you will see that in that case, it was oxygen only, but I told you it can be oxygen, nitrogen, or chlorine. So in this case, if you see the thymine, okay, and then there's a oxygen, double bond with oxygen, and this is a hydrogen bonding. So bond between hydrogen and oxygen, and there's a nitrogen on the other side. So basically you have a NH2. So one H is making a bond with because of N and H2, the hydrogen will have a slightly positive charge, and this will have a slightly negative. So you see the hydrogen bonding. So everywhere, wherever hydrogen bonding, you will see between H and O, or H and N. So for example, in this case, this is H and N. This is H and O. This is H and N. This is H and O. Like that. Okay. So this is hydrogen bonding. And so good thing about the hydrogen bonding is that actually you can increase the temperature. You can break the bonds also. At certain temperature, the DNA can dissociate into a single strand. That's possible. Okay, let's move on. We already discussed about 3' and 5', why we call it as a 3' and 5'. So whenever you write a sequence of any DNA, so make sure you always mention 5' and 3' ends, so that we know which direction it will elongate during a PCR. Okay. And the structure is called a double helix, which we already mentioned. I think it's fine. So let's move on. Now some sizes of a DNA which are also important. In this case, if you look at the structure, so the DNA structure is such that it has some minor grooves. So if you look at this structure, so this is a major groove, right? If you call that V as a groove, then this will be a major groove, and then there is also a small groove here. That's called a minor groove. So the major groove, the number of bases between a major groove are about 10. So there are 10 bases from one point to the other point, the same point. It's repeating itself, the structure. So from here to here, it is actually 10 bases, and the total distance between 10 bases is 3.4 nanometer. Okay, so 10 bases are equal to 3.4 nanometer. It means between each base, the physical distance is about 0.34 nanometer, right? So now there is a question for you. You know how long is a human DNA? We discussed it one time. Huh?
Very long.
Very long, yes. I think if I'm not wrong, I think it was 3 billion. I'm confused between 1 billion, 3 billion. I think it's a 3 billion base pair long, approximately. So if it's a 3 billion, so it means 3, let's do the quick thing, so that we can do the right calculations.
It was like 3 meters. Yeah.
Length of 3 meters.
Around, yeah. Or it'd be 3.4.
It's a 3 billion, approximately 3 billion bases, okay? So it was 3 billion. Okay, so 3 billion means 3 into 10 raised to the power. How much? 3 into 10 raised to the power? 9. Okay. So 3 into 10 raised to the power 9 bases. Can you calculate how much would be the length of it? Anyone with the answer? 1 meter? Yeah, approximately.
It's in the helix thing, right?
Yeah, but if we twist them, but so it's approximately 1 meter. Okay. I didn't do the math, so I'm just telling the answer. I don't remember what was the right answer, but I think it's 1 meter. Okay. So 1 meter long. Okay, and then the thickness of a DNA, if you really grab it in your hand, how much thick would be from a side? Say it's about 2 nanometers. So in reality, I think it's about 1.4 nanometer, but I think for simplicity, let's put it 2 nanometer approximately. Double stranded. So it means if it's single strand, it would be about 1 nanometer. Okay, and so let's say now if we want to read the DNA for many applications. So if we can read the DNA, we can solve many problems. For example, we can read genes, we can do genetic analysis, we can sequence the DNA, we can identify people, who they are, right? It can have thousands of applications in forensics, in health sciences, you name it. And then it is a code for a lot of actually diseases also. We can find, okay, this is, but the problem is how DNA is sequenced. So like 10, 15 years ago, that was very time-consuming process. You have seen in the video, right? They have to cut a whole DNA segment into small sections, and then each section, then they copy it to make a lot number of similar copies, and then you put some bases with some fluorescent dyes, and we'll go through it briefly. I think I'll explain you one more time. It's called Sanger's chemistry, and then you actually run it through a gel, and then with the fluorescent you can detect it. So it's a long process, and you cannot really sequence I think more than 200 bases at one run. And if we have to now sequence 3 into 10 raised to the power 9 bases, I don't know how many runs you have to do it to sequence the whole segment. But now there are new sequencing technologies are now out, and which people are using, and they have significantly improved the throughput and speed of the sequencing. The RNA, let's say if we are take a blood sample, if we want to detect if someone has a disease or not, normally we detect it with the PCR. We look for specific segment of RNA or DNA in a blood, and if that is present, we consider, okay, this person might be infected with the virus, for example, or bacteria. But the other thing is, if we have such a nice device that can sequence the DNA very quickly, we can instead of looking for the genes, we can even sequence the whole RNA, and that would be very specific. But it's not very cost effective so far. But anyways, the one way to sequence, because there are now dimensions, it's a 2 nanometer, so all of a sudden it came to my mind, okay, why not we can have actually, we can use a nanopore, and I thought it's a better, maybe good place to discuss about the nanopore briefly. I think one question was also in your homework about nanopore, so you might have some understanding about what is a nanopore. So in this case, if this is a 2 nanometer and we want to read the double-stranded DNA, or let's say if there are some segments of double-stranded DNA in a solution, and we want to just find out, okay, how much is the length of that double-stranded DNA. So what we can do is we can maybe run a small chemical cell. Let's say, so let's say we have a one chip, which is a pore, and the chip is we put it in a chemical cell. Where one side we put a negative electrode, on the other side we put a positive. This is negative, this is positive, and now there is a pore here. So this pore, which is very small, it can be about very nanometers, and this whole thing is filled with electrolyte. Okay, but this chamber and that chamber is isolated from one another except the pore. The pore can connect them. So it means if there is any current flow, it has to be through the pore. Otherwise, it should be super packed. There should not be any leakage current. So if you apply some voltage, let's say if you apply 1 volt. So what kind of, let's say if this is a voltage, you're applying, how much you're applying? 1 volt, right? Continuously. And this is your current. What kind of current you're expecting? Like falling down, increasing, straight line, parabolic? So what kind of shape of a current you're expecting from this experiment? Just electrolyte.
For more voltage, more current.
More current, so it should be just a 1 current value, right? Let's say if you apply 1 voltage, you might get 2 microamperes, for example. I'm just making up this number. But that will be dependent on, so let's say we'll be getting this current. Okay, some current value, like you are measuring the resistance of anything. Now, let's say if you change the conductivity of the solution. Previously it was 0.1 molar. Now let's say you make it 1 molar. So you make it more conductive, for example. So then what will happen? So if you apply same voltage, will you get a current here or there? On the top or bottom of the previous line? Above. That's good. Now, so it means we can measure the actually conductivity of different solutions by using this setup, correct? And we can also measure the size of the pore also, if we know the conductivity, right? Because the conductivity will be dependent also on the pore size. And if the pore size is small, it will not allow a lot of ions to pass through very quickly. So your conductivity will not be the same as your bulk conductivity. Bulk, when we say, is when you don't have this pore and you made a voltage and the electrons, the ions are free to pass through. But in this case, there is a pore there which can restrict their movement. And up to a certain level, it restricts their movement. Otherwise, if you increase the size, let's say 100 nanometer or higher, then it becomes, its conductivity is equal to a bulk conductivity. I think I'm going too much detail. But so the point was here that now let's say if I want to put a DNA and I want the DNA to actually pass through this pore. So which side? So this is the right side, okay, and this is the left side. So where should I put a DNA so that it can move through the pore? Towards the right side or towards the left side? This is right, this is left. Left side. So I'll put a DNA on the left side actually, right? And the reason is that DNA has a negative charge, and so that it can move through. Because if you put it here, then you'll not see anything. It will just stay there. Maybe it will just stick with the electrode. So if you put it on this side, and now if you apply voltage, let's say 1 volt again. So the negative, again, the negative charges, let's say previously, let's say if it was a KCl, for example, K is a potassium and Cl is a negative. So the negative charges were moving towards the positive electrode, and positive potassium was moving towards the negative. So that's how electrochemical cell works. So now we have molecules which are, now in this case DNA, it can be also protein, which also have charges at certain pH. So these DNAs actually will pass through the pore to go to the other side. So when DNA is passing through the pore, what kind of current you expect? Will you again expect a straight line as before? or there will be some dips maybe, right? So let's say I'm just drawing it in the middle. Doesn't mean that it will be in the middle, but just drawing it for using the space. So previously, let's say there will be some current, and after some time when DNA started passing through, I have seen there are peaks like that. Okay, which you will see. I think it's better to show you also the peaks right now. For example, these ones, okay?
You're coming this way.
Oh, okay. Sorry. For example, these ones. So this is the real data, okay? But the thing is, this value is very small, 2 nanoamperes. So because nanopore is very small, that's why the value for current is very, very small that you're measuring. And if this is 2 nanometer, let's say the height, so there's a significant noise, right? Still the signal is about 5 times more than the noise in this case. But sometimes you get a very bad signal. So it's very difficult to differentiate whether it's for example, this is also nice, but sometimes it's difficult to really see whether it's a signal or noise itself. The equipment has to be super stable, isolation-free table to record these readings, and then you also have to put the whole thing in a Faraday cage. So Faraday cage is nothing, just a metallic box where the optical light and other radiations will not affect your experiment, and I've seen that happening. If you don't put it inside a Faraday cage, if you don't put it on an isolation-free table, your noise is significantly fluctuating. And that noise comes from the equipment, like when it's moving, so small movement can actually cause a big problem in nanoscale. So then it becomes very important when you're taking readings in nanoamperes or picoamperes. So I'm going back to our dock camera. So basically you're expecting this kind of a behavior, right? Now, the thing is, if one DNA is long than the other one, so what do you expect? So we expect that in that case, we might have maybe a longer dip, right? So it means by looking at the shape of the dip, we can identify if DNA is long or not, and from the ratio, we can understand what is the approximate length of a DNA, right? So that information we can get very easily. And it's very straightforward. It's very easy to operate equipment, the setup. So as compared to, we don't have to use any fluorescent dyes. There's nothing fancy in it. It's just putting the DNA negative side passing through the nanopore, and you get a signal. But on the other hand, can we really, when people started looking into nanopore, the goal was actually to really identify whether we can sequence the DNA or not. So we really want to read the DNA when it is passing through base by base. And so thoughts are that when DNA is passing through, it's not straight. So each base will interact differently inside a pore, and then you might see some difference in a signal, and that in that way you may be able to identify whether it's A or T or G or C. So that's the goal. And actually, not like that, the one DNA is passing through, but by using nanotechnology now, people are able to sequence the DNA very recently. And in the one product is already out there in commercial zone. If you want to look at Oxford Nanopores. Oxford Nanopore is a UK-based company, and they are selling their device for sequencing. They call it as a home sequencer, so it means you can use it anywhere, but it's not that straightforward to use it. You need to know a lot of things. But basically, the setup is very small, and they have shown that they can sequence the DNA through this. And there are some limitations which we'll talk about when we talk about DNA in a little bit more detail, the nanopore with more detail, like why sequencing with the nanopore is challenging. and why it took so long to sequence the DNA and why there are error rates. So there are reasons for it. But for now, they have shown with the 90% reliability that they can sequence the DNA, but still there are some errors in reading the some sections of the DNA. But so in this case, let's say let's take another example. So it means we can really measure if the DNA is long or small. But the experiments can be designed this way. People have shown it previously. Let's take one example of oncogene. So some of the cancer, the reason is just one base pair mutation. So normally it's the whole gene is mutated or changed for unknown reasons, and that's why your cell behave differently. And sometimes it's not a single gene; it's a multiple genes which are mutated, and sometimes some of the cancers are just or diseases are just because of one base pair change. So let's say if there was A somewhere, now it is a T, for example, or G, whatever. So that mutation caused the gene to function differently, and now cell becomes actually cancer cells. And oncogene, when we say oncogene, means the cancer cells because of a single base pair mutation or single gene mutation. So so there was one device which was proposed based on the nanopore for detection of oncogenes. So it means if we can identify from a sample which may have perfect complementary or not perfect complementary, or like the wild type gene which is like real gene sequence, and then there is a mutated gene which can be cancerous, like where one base is different or two bases are different. And if we can identify that this is different than that one, so it means we can really tell that if this gene which is present is a mutated one or is a wild type, means if it is a healthy one or the cancerous one. So what we can do is, what they have done is they use the complementary properties of the DNA, and they have built actually a nanopore. I'm just showing you nanopore now, a big one, and they put some strands inside, functionalize it, okay, with some strands, and then they designed experiment where there was actually one segment of, and there was another, and let's say this was A and this was B. So A was, let's say this is A prime, okay. So A was healthy gene which has some sequence, and then B was exactly the same gene except at one place one base was different, okay. So instead of T they put G or A, okay, only one base was different. And now they moved this DNA through this pore by using a similar setup, and they noticed that when this DNA passes through, which is a perfect complementary to the other side, means they will show strong binding, and they have seen that DNA actually moves inside very quickly and passes through it because of they have noticed this behavior because it's complementary, so it may try to attract it, or it allows it to actually come inside a pore rapidly because it's a complementary opposite charges, for example. And they were seeing the pulses which were actually shorter, okay, when that segments are passing through. And now when the non-perfect complementary, or the segment which has a one mutation or one change in the base that passes through, they notice that this pore doesn't allow them to pass through very quickly because it's the opposite, so it actually doesn't show any interaction, so it takes some time for the DNA to actually go inside the pore, and they noticed that it was taking a little bit time. So this is just a drawn not to scale to just show you that it was taking a little bit more time to pass through, and from those pulses they can tell. And now it can be unknown sample, okay, but they know these are the pulses, so we can really identify if we can isolate that DNA and then maybe cut that specific segment of a gene, and now we can, instead of sequencing it, we can test it with this device very rapidly whether this is cancerous or not cancerous, for example. Okay, so this is one application they showed previously. So it means you can do many different things with the nanopore. And yeah, but do you think what type of sizes would be used for nanopores for DNA experiments? Greater than 2 nanometers, yes, so about 5, 10, but not 100 or not 200, because if you use such a high diameter, then when DNA is passing through, you may end up not seeing actually anything in your current. Because let's say if the pore size is 200 nanometer and DNA is, let's say, 2 nanometer, if that is passing through, how much of the pore it is blocking? 1%. So 1%, if you look at the noise, it's even more than 1%, it's about 20% of the signal. So it means you will not see anything theoretically, and that's what you will see actually also. So it has to be really small. But if you're working with the proteins, for some applications, you want to characterize proteins, their movement, their binding, and all that. People have shown it that they can do it with the nanopores in the educational lab and research labs that that can be used for that purpose. So I noticed that for proteins, you need a larger pores. It depends on the protein size. So you can use the pores of 100 nanometer because some proteins are 50 nanometer, 20 nanometer, in that range, and it can show you larger binding signal. So understanding the DNA sizes are important for some applications. The charge is also very important. So these are the people who actually were behind the discovery of a DNA structure. Rosalind Franklin was the first one, as per my understanding. In 1952, she first time used an X-ray diffraction, okay, to to find out the crystal structure of DNA. Means previously people don't know whether it's how the ATGCs are oriented. It's a double helix or single helix or whatever. So it's not that far. It's not that part of history. Still, it's 70 years old information, right? So it's very recent development. And then the other two people, Watson and Crick and James Watson, Francis and Crick and James Watson, they explained that helix structure in 1962, and then they were 1953 later on, and then they were actually able to get a Nobel Prize for their work. Some people have different opinions. They think maybe Rosalind's work was more pioneering, and she should be getting the Nobel Prize. But anyways, but Nobel Prize is not everything, right? It's just one encouragement, I would say. But some people might have done work which is more than what Nobel Prize people have done, right? So, it is important if you get the encouragement or some prize, that's good. But if you don't get it, it doesn't matter. Now, so they use actually she used X-ray diffraction to explain the structure. So this is one image I just want to show you how the X-ray diffraction work very briefly. So basically, you have an X-ray source, and when we say diffraction, is actually scattering of light from a molecule or a cell surface or anything. You can have also single pinhole. I think we talk about the diffraction when we are discussing photolithography, right? So I'll not go into those details again. So what they did is they put a crystalline DNA, it means a dry DNA inside on some surface, and then they shine an X-ray beam on top of it. And then they arrange the DNA crystalline in certain orientations. They all were actually parallel to each other, all strands. And they were able to see some very distinct pattern of dark and bright fringes, which you also see when you have small pinholes and you get a diffraction from there. And that pattern was showing the structure of because the scattering was showing the structure of the DNA, and then later on it took them some time to explain how it is oriented, but they were able to actually figure that out. Okay, this is how it is oriented. And the X-ray diffraction crystalline structure is commonly used to actually investigate the crystalline structure of the DNA and also other proteins. Very commonly, if there's a new protein and we don't know how the structure looks like, so they use X-ray diffraction to investigate how those protein folds and all that. So it's a very good tool actually, but it's a significant. It's not like you put a sample and you will find out, okay, what is the structure. So sometimes it takes 5 years, 10 years. It may be one PhD thesis or a couple of PhD theses to explain one structure. So it's a complicated process, but that's what they use for explain the structure. So now let's say if we have one, we know what is DNA, right? And what it is made up of. So if I ask you a question just for revision, what is the difference structurally between DNA and RNA? Instead of thymine, it has uracil. That's the one thing. RNA is a single strand, but DNA can also be a single strand, right? So there are some single strand DNAs.
RNA can't form double strand.
It can form double strand. It can, yeah, with the DNA, it show binding. It can form double strand with the DNA, single strand DNA.
No, okay.
No, but in reality, in the nature, you might, but you will also see some RNA folding. That when there are RNA such that it is complementary to some sections, it will show some folding. So it's not like, but it is normally not present. The purpose is that when we want to take information from inside to outside, it's normally single strand. That's fine, but it can make double strands. It's not like it cannot. Huh? Hydrogen missing. It's deoxyribonucleic acid and ribonucleic acid, right? So there is a one oxygen missing, deoxy. So it's actually one oxygen missing at group 2 at C2 location. So in one of the pentose sugar, there was because one case we are using deoxyribonucleics, and the other case we use ribonucleics. So the difference is on the deoxy, which is DNA. So there is a one oxygen missing, and I think C2 location. And instead of that, I think we have a hydrogen there. And that's important to understand because later on that defines why DNA is more stable in a solution and why RNA disintegrate very quickly, and that's why it's very difficult to work with very sometimes because it has oxygen there, so it reacts with the chemicals more vigorously, so it can actually break down very quickly, and that's why people don't like to work with the RNAs. So what they do is normally they convert RNA into DNA. There is a process, and then they work with the DNA because DNA is more stable, considered more stable than the RNA because of that just O thing there. Okay, the DNA microarrays. So before I jump to microarrays, I briefly want to talk about where we were on when we watched the video for Human Genome Project. Basically, at the start, they use a Sanger's chemistry. So that's a one basic method which people use in analytical chemistry labs or biolabs to sequence any DNA, and that is explained there. So let's go over that Sanger's chemistry briefly, like how that works. Although it is in a video, but I noticed that it may be a little bit difficult to understand the whole video. So I thought, okay, maybe it's better to go over one simple example so that you have some understanding about it. Okay, so let's go to document again. So what we'll be doing now, we will be sequencing a DNA. Is it focused? Okay. So let's say we have a sequence which is 5 base pair long, 1, 2, 3, 4, 5. I'm just taking a simple example. It's a 5 base pair, not long, so it can be 20 base pair, 30 base pair. And let's say it is a AAG G, C, and T. That's it. This was a sequence, unknown sequence. And now we want to decode it. And so in reality, let's say if a long DNA, so you break it into small segments of 200 or 300 base pairs, and then you actually multiply that with the process, grow in a bacteria. So, but then you have a lot of same sequence, but everything was 200 base pair long. Like same segment was grown over a period of time, multiplied into thousands. So, so let's say we have one of that sequence, which is now in this case is 5 base pair long. So the technique was based on the fact that DNA bases actually bind with the complementary bases. So what they do is, now let's say in a solution we have a lot of bases freely available nucleotides. Ones are A's, we have T's also available, we have G's, we have C's, a lot of them. So now I'm just drawing one, but in reality consider this is one 5,000, these are 5,000 sequences, and these bases are maybe 10,000, 20, 50,000. So there are a lot of them are available to you, okay? And then in a solution, and then we also add some bases which are actually same AT, GC, but they have also some fluorescent dyes with it. So let's say A has red, for example, dye which shows fluorescence when you shine it with laser, the red laser, and then this may have a, let's say, blue, RG, green. There's okay. Let's put green with the G. Okay, and then C, yellow, for example. Okay, so let's say now same bases, but they also have some colors. But not all the bases have color. Some of them have color. So now if you put them there, so what happens first? Let's say this base come in contact with the one of the strand. So this is C. Will it bind there? No, it will not bind there, right? So then it will go off. Then maybe A, G, or maybe A go there, it will not bind. So when T goes there, right, it will bind there. Then the second one, if we have a T there, it will bind there. Another T, but if some places we, let's say, get this T, which has a blue color, if that goes there, it will bind. This is blue. But those bases are designed such that, which have fluorescence, that it will not allow any further elongation. Okay, so the reaction will stop there whenever the color, any color base goes and binds with there, so it will stop there. So with this strand, it will stop there. Okay, but now we have other, like I told you, 2,000 different copies of a similar strand. So let's say we have another one also available, which is again AAGCT. So in this case, there is a possibility that in the first place we might have this T with the blue. So this will stop there right away. So there are some sequences where randomly it will stop at the first place. There are some sequences which will stop at the second place, and there will be some sequences which may go all the way. Let's say AAGCT, which may go all the way AT, ATGC, CG, and TA, which may all the way without having any fluorescent base. And let's draw all the combinations. What can be possible? AAGCT. So let's say now we have one in second place. This is TT, regular T's and C's, the G, and let's say if the C comes, which is with the yellow. Okay, so this will block there, and there will be two more. 1, 2, 2, 3, 4, 5. 1, 2, 3, 4, 5. AAGCT, AAGCT. Now we have a TTC and then CGC, right? So this will be G with the green. It will stop there, and then we have a TTCG, and this one would be A with the red. So these are 1, 2, 3, 4, 5, 6. So 6 different types of double-stranded DNA strands will be formed, right? So without base, it allows full elongation. Some places we have when fluorescent dyes, bases with the fluorescent dyes may come into place. Now, if you have a 5,000 similar strands, so maybe 800 you get one type, 800 you get second type. So there will be randomly different variations of those strands available, right? But these are the sum of the variations. These are the only variations which are possible, right? So nothing else. Now the problem is if you have 200 bases, then you will have 201. So in this case we have 5 bases, and how many different variations we can get? 6. One is without any dye. Let's say now if you have 200 bases, so you will get 201 different lengths of DNA formed. Okay, now let's say we have this process is all automated, so you don't have to really do anything. Just take a segment, put it into the bases, and let it incubate for some time, and these structures are automatically made. Now, how we can see now they decode the sequence. So there is a process called the gel electrophoresis. So electrophoresis is the DNA. When we were talking about the nanopore, actually nanopore is also called the electrophoresis process, where you apply voltage to different places, and there is a solution in between, and then the molecules move from negative towards the positive side. Okay, or positive negative. So movement of the molecules under the influence of electrical potential is called electrophoresis, and that's why the nanopore technique is called also the electrophoresis technique. Okay, but in gel electrophoresis, it's clear from the name, instead of electrolyte we have a gel. So let's say if we in nanopore setup, instead of electrolyte we put it in a gel, then it will become a gel electrophoresis. So, but we don't really need a nanopore there when we run a regular gel electrophoresis. It's just a gel, and you put the DNA to one side, you apply some potential. So let's make a gel. Let's say there is a gel. Gel is like a jelly which you eat, gelatin, for example. If you look under the microscope, it's a porous structure. It's just gel structure, porous, long chains of polymer, and there are different types of gels available. Agarose and, for example, is the one commonly used type for DNA work. And you can mix it in water at different concentrations. It will make gels more densified or less densified based on how you work with your gelatin at home. So it's very similar. So let's say this is a gel. I'm not drying it. I'm just leaving it empty. But there's a gel there. And then let's say this is a negative terminal. Let's say this is positive, for example. And now you put your DNA, this whole thing which was created, you put everything there, okay, in small inlet, like this place, all the solution. And you apply voltage. So what will happen? All these bases and structures will try to move towards the positive side, right? When you apply voltage through the gel, and when they move towards the positive side, the ones which are smaller in size, actually they can move further in the same time. One which are lengthy, right? It will take them more time to pass through the gel under the same voltage effect. So once you put them in a gel, you will see that there are some bands created. And those bands are created based on their sizes. So let's say if one DNA was small, it will be able to move much further towards the positive. If it is a larger segment, then it cannot move very quickly. It cannot travel a large distance in the same voltage conditions through the gel because it's lengthy. So you can resolve that gel. It means you can see even a single base pair differences if you run a very good gel. So and the thing is, it's not only one strand. You will be at this place, there will be how many of similar sequences? About maybe 800, for example, in my examples, there will be 800 similar segments of a DNA will be here, 800 ideally in this place, that place, that place, that place. So the signal, fluorescent signal will not be coming only from one segment. It will be like 800 similar segments. So like blue, it will be 800 blue, 800 yellow, 800 green, something like that. And if you have this, and if I read this under the microscope, I can tell, okay, this will be A, this is blue, for example, or this is red, for example. This will be blue, right? The first one is a T. So first one would be actually blue. Yeah. First one will be blue because this is the smallest one. Correct? So this will be blue. The other way is that you can actually let them pass and you can put a laser here. Okay, let's put it here a little bit. So there is a laser. So whenever these bases are passing through, in that order, based on their length, so let's say the first will pass through, you know that the smallest one is this one, right? Because this is the smallest one. You only have one complementary base. So you will get blue color. Okay, let's say this is a detector. So you get a blue, for example. Okay, this passes through. The second passing through, you will be getting blue again. Then the third one will pass through, which will be this one actually, and you will get it then yellow signal. Then fourth one, you will get green. And then fifth one, when like this one will pass through, you'll get red. And when this will pass through, you'll get nothing. So you will not detect it. So we know that blue was attached with what? T. This was also T. Blue is T. Yellow is what? C. And then green is G. And the red is A. So this is a sequence in which they are detected. And we know that it will be, original sequence will be complementary to this one. Right? We know that already. So it means that will be complementary to this sequence. So, and that would be T for A, right? C for G, and G for C, and A for T. So this would be our original sequence. Is the same? AAGCT, AAGCT. That's correct. So it means we have sequenced the unknown segment of a DNA by doing this. And it can be 100 base pair long or 200 base pair long. Right? So now it means if it is a long segment, so we can cut in small segments and then we can do this process over and over, and we can then start resolving the sequence of a DNA. And that was done in that video, and it was taking so much time. Okay, but let's move on now. Okay, so, but there are some arrays of DNA segments, which, let's say we have a surface where we actually put different DNA segments, and then we let the sample to actually incubate with it. That we call it a DNA microarrays. Micro means the micro spots of different DNA structures are created on the surface. So now, and they find a lot of applications. So in this case, we don't need to sequence a DNA. Let's say if we have a segment of a DNA, and we are only interested in specific, if that segment of a DNA has specific gene or not. Okay, so what we can do is, one way is we can sequence the whole thing, which may not be time efficient. We can maybe take a DNA microarray setup, which will have actually thousands of different microspots created on the surface with the technology, the lithography or other methods. And now you put a whole sample on top of it, and you will see that where the sample which you're putting in, where that sample is binding, which places on which spots. And you already know at what spot you have what strand of DNA. So let's say if it's binding with some place A, you know that at that place we have this sequence of DNA which we created there, which we actually immobilize on the surface. And then it also showing binding at the T for another location, not T, let's say Y. And at a place we know this was a fragment of a DNA which we already put in there. So in that way we know that what kind of bases are there in this sample. Okay? I'll show you one example in a minute. And then, so basically DNA microarray is just a glass slide, or it can be another material which you functionalize it. And then you attach an oligo. Oligo is single-strand DNA, okay, where you know the sequence. And you have multiple of those places on the surface. And then the next step is actually putting a sample. Let's say then you put a sample. Let's say this is your sample, which you put on there. And you will see that it will bind at different places. And when it binds, but in DNA microarrays, when you create a microarray, you already know that in 1,000 different spots, what are different sequences which are present there. So each place, your I call it oligo size is about 20 base pair long. So each place you have 20 base pair long segment. The other place will have a different 20 base pair long. The other segment will have 20 base pair long different. So let's say if we have a 20 base pair long oligo, how many different combinations of oligo we can have? Question for you. If we have oligo, which is just a single strand DNA, another name for it, but it's just a single strand DNA, and there are 20, it is 20 base pair long. Okay, 20 bases long. Then how many different, how much? To the power 20? Okay. No, that's not correct. 20 power 4? Not correct. 4 power 20. Okay? So it will be 4 power 20. And now you can do the math at home. How big number that would be. So basically, in order to cover all different sequences in 20 base pair long oligo, you need to have 4 power 20 different spots on the microarray, and that will cover the whole variations of 20 base pair. And that's how it is created. So it's not a very cost-effective process. It's very expensive. So I'll give you one example here. So let's say we take a tissue sample, and we want to sequence some RNA from that tissue, for example, okay, for some reason. And the tissue is green. It's just showing it that we isolated the RNA from those tissue samples. Okay, tissue is some segment, some part of our body, or some heart, or some other place. Then we separate the RNA with the process. Okay, which we're not going to tell how to separate RNA from a sample. Just leave it as it is. So, but we get RNA from 2 different samples, and then before putting it on a microarray, what we do is we tag them. So let's say one sample, which is sample A, we tag it with a red dye, and the other sample we tag it with a blue dye, for example. Okay, and now we put both of the samples on the surface and let them incubate for some time. And you will see that this mRNA, which is red, it will show binding based on its sequence, because it may be 200 base pair long, it may be 1000 base pair long, but wherever it will be complementary, it will show binding, it will bind only on those spots. So one mRNA which is long, it will bind not only at one place, it will bind at multiple places, right? Because the oligo which is complementary to it is only 20 base pair. But then mRNA, it may be 1000, 2000 base pair long. So it means wherever it has that segment of 20 base pair oligo, it will show binding there. So basically, this red and blue, both of them will bind at multiple places. And then we'll also see that they bind some places, they bind together at the one place, where we know those are the common signals. So it means that segment is common between mRNA 1 and mRNA 2. And that's why you will see the yellowish there, like blue and red both. So in that way, you actually then scan the whole 2.1 million different sequences, like 2.1 million different spots. But scanning is not that difficult because you do that with the image processing. You scan the whole surface and look at where is red, where is green, where is blue, where is yellow. You just look at it, okay, where it show binding, and you already know that what sequence was you already immobilized of the LIGO. So from there you can do the reverse engineering, you can find out the real sequence. Okay, you can find out what was the real sequence there, and you can also find out, okay, what is common between mRNA1 from A or mRNA B, because if they are showing binding in one place, it means those segments, they are showing a common region. Okay, so in that way, microarray can be used for many different purposes. There are also some protein microarrays available also, in that case you can have different spots where you can functionalize different proteins, and then you put a sample and then see where it's showing binding. So another way, but you have then many different spots, so you can read it very quickly. I think that's we kind of talk about that under fluorescent light, color generated and the genes can be analyzed and all that. I think we covered that. We still have, I think, 25 seconds. I think I'll stop there. It's not good to move further because, by the way, I don't know whether it's a time for announcement or not, but basically there will be, I'll be traveling most probably on Thursday, and then I'll upload the video lecture for that. So I will not be, there will not be class on coming Thursday, but I will upload the lecture for all of you.